Hello, everyone. Thank you for joining our webinar today. We greatly appreciate your time. My name is Kristen Pauly. I am the Director of Marketing and Business Development here at ImmunoChemistry Technologies, and I will be moderating our webinar today. Your phone has been muted. However, we encourage and welcome your questions. Please type your question into the chat dialog box on the right side of your screen and select Send to Host. If you have any technical difficulties during the webinar, please send your chat to me. It is my pleasure to introduce our speaker, Tracy Murphy. In this webinar, Tracy will share information on fluorescent assays for in vitro imaging in cancer research. Thank you, Tracy.
Well, thanks, Kristen, for that introduction. And thank you, of course, to all the listeners taking the time to join us today. Also, in reference to the cancer topic, we did just get back from the AACR conference, which was last month in Chicago, where we had a lot of great conversations and a lot of interest from researchers who are conducting this type of work. So if you're tuning in today after visiting with us at the show, welcome. Thanks for tuning in. To introduce myself, I'm the Director of R&D and Quality Control here at ICT. I work very closely with our R&D team to develop new products and applications and to keep things launching on our website. As we move forward, in addition to any questions that you have on today's presentation, if you have any ideas or suggestions for future products, you can also feel free to chat those in as well during the webinar. Today's talk will be focused on looking at how some of the tools that ICT offers for fluorescent imaging can be applied in the context of cancer research and oncology. So some of the things we'll be touching on, we'll learn how to quantify caspase activity in vitro, how to measure cathepsin activity, detect mitochondrial membrane potential and depolarization, differentiate necrosis and apoptosis, measure serine protease activity, and finally how to detect pyroptosis. We'll also be discussing the utility of this technology in the current cancer research landscape. To give a little bit of background on ICT, we're located in Bloomington, Minnesota. Bloomington is a suburb just south of Minneapolis within the greater Twin Cities metropolitan area. ICT was first founded in 1994, so we've been in business now for over 23 years. Here at ICT, we do offer both products and custom services. In terms of our products, we manufacture high-quality biomedical research reagents, including unique cell viability and apoptosis assays, as well as proprietary ELISA reagents. I'll be providing some more information on these offerings shortly, but I did want to note that all of our products are for research use only and are not for use in diagnostic procedures. When ICT was first founded, we actually originally began as a services company. So we do have years of experience with protein chemistry and ELISA optimization. We can help you develop reliable, sensitive, and specific immunoassays. We can also scale up and manufacture an assay, so all of the components would be provided in a ready-to-use format. If you're in need of a service project, feel free to reach out and get in touch with us. Our skilled team is happy to discuss your needs, and we do offer everything from initial consultation, support, to start to finish management of your project. One product line that I just wanted to briefly touch on today is our ELISA reagents. These include all of the components that you would need to build a better ELISA. Our coating buffers, blockers, sample and assay diluents, conjugate stabilizers, substrate stop solution, and wash buffer all work together to improve assay performance at every step. These products have been specifically designed to address common issues that are frequently encountered during ELISA development. To summarize, ELISA development really is a long-standing core specialty of ours, and we're happy to work with you to help you optimize your immunoassays. That's all I'll be mentioning about our ELISA products, but if you have questions about this product line, you can feel free to chat them in, and I can get you a response after the webinar. The products that we'll be focusing on for today's presentation is our cell viability assays. So these assays include a broad range of fluorescence-based whole cell assays for intracellular apoptosis detection and cellular analysis. ICT's line of assay kits can detect apoptosis, necrosis, autophagy, pyroptosis, cytotoxicity, activated serine proteases, oxidative stress, mitochondrial membrane potential, and much more. Our kits are designed for use in whole living cells, so no lysing of the cells is required. Some of you might be longtime users of our popular FLICA product line for caspase detection, but we do offer so many more things in a wide range of fluorescent applications. So in the webinar today, we'll be covering several different of our fluorescent detection reagent product lines, and they're listed here. FLICA for the detection of active caspases, magic red substrates for the detection of caspases and cathepsins in real time, MitoPT for the detection of mitochondrial membrane potential and depolarization, cytotoxicity. Our kits are designed to measure cell-mediated cytotoxicity, so this is where cells of the immune system lyse and kill infected cells. FLISP for active serine protease detection. We'll also look at an assay for measuring necrosis versus apoptosis. And finally, we'll finish with a discussion of pyroptosis, which is a form of inflammatory cell death, and we'll highlight a detection assay that we have for measuring it. As we work through these main key topics, we'll also discuss some of the cancer research applications of each of these different product lines, and we'll be reviewing some sample data. So I'd like to begin today with a discussion of FLICA. FLICA is one of our most popular product lines here at ICT. FLICA stands for Fluorescent Labeled Inhibitors of Caspases. ICT's FLICA kits offer an in vitro whole cell detection method to study caspase activity in apoptotic cells. Our FLICA assays are available for a variety of different caspases, as well as polycaspase activity. And the samples can be analyzed using a flow cytometer, a fluorescence plate reader, or a fluorescence microscope. Our FLICA kits are available with green, red, and far red fluorescence options. Cancer is a large class of diseases involving abnormal cellular growth with the potential to invade or spread to other areas of the body. Cancer cells grow and divide without being directed to do so, and they continue to replicate even after being signaled to stop. So cancer can be thought of as a scenario where too little apoptosis is occurring, and this is resulting in malignant cells that will not die. Therefore, when it comes to cancer, apoptosis can be viewed as part of the problem. However, it may be part of the solution as well, as apoptosis plays an important role in the treatment of cancer and is a popular target for many cancer treatment strategies. So in terms of cancer research, there are several advantages of using FLICA over other methods. First, it's the most accurate method. It's measuring the actual intracellular process of apoptosis via caspase activity, instead of just a side effect, such as, for example, the translocation of phosphatidylserine. Secondly, it is the most sensitive. Our FLICA kits allow you to detect cells in early apoptosis and differentiate them from cells in late apoptosis. So in flow cytometry, this will often appear as a bimodal or a two-peak curve, consisting of fluorescent early-stage apoptotic cells and then even more brightly labeled cells, which are in later-stage apoptosis that have even more caspase activity. So this kind of sensitivity means you can see how your cells are responding to the treatment. So whether they're unaffected or they're just starting to become apoptotic, or perhaps a more robust apoptotic response has been induced. Finally, we do have a number of different types of FLICA kits available depending on your cancer research goals. Our FLICA kits are available with varying caspase targeting sequences. So to assess apoptosis in general, we do have our FLICA polycaspase assay kits. But if you'd like to look at a more specific caspase or a cell death pathway, we do have kits available for detecting caspase 1, 2, 3, and 7, 6, 8, 9, and 10 as well. Let's take a minute here and look at how FLICA works. First of all, our FLICA reagents consist of these three key components. So we have a fluorescent reporter tag. This is the FAM or the SR or the 660 fluorescent label. We have a caspase targeting peptide sequence, such as VAD or valine alanine aspartic acid. This is shown here. And then an FMK reactive group. The FMK reactive group is what actually forms a covalent bond with the catalytic sites of the active caspase enzyme. So this is what enables the FLICA probes to be retained inside the cell despite the subsequent wash steps. Once the FLICA reactive groups once the FMK reactive groups on these FLICA probes form these covalent bonds with the catalytic cysteine groups within the caspase reactive sites, this means the caspase enzyme becomes inhibited, so it's no longer able to cleave any substrates. So now that we've talked about how FLICA works, let's examine some sample data of FLICA in action. So here we have some sample flow cytometry results. Now in each of these overlay histograms, the red line represents the healthy control population, and the black line represents the induced experimentally treated population. So in this first panel, the Jurkat cells were treated with staurosporin to induce apoptosis, and then they were stained with our far red polycaspase reagent, 660 VAD-FMK. And we can see a clear shift along FL4 in the treated population. This panel also highlights the sensitivity of FLICA. So as we just discussed, here we see a nice example of that bimodal two-peak curve within the positive population. So these two peaks represent the early apoptotic and then the brighter later stage apoptotic cells with even more caspase activity within the sample. The middle overlay histogram shows the results when staurosporin-treated Jurkat cells were labeled with ICT's green caspase 3,7 reagent, FAM-DEVD-FMK. And these results again show a shift, but this time it's in far red fluorescence along, instead of being the far red fluorescence along FL4, we see it's a shift in green fluorescence along FL1. Finally, the last panel on the right shows THP1 monocytes, and these cells were treated with LPS for 3 hours, and then they were stained with our far red caspase-1 detection reagent, 660 YVAD-FMK. We can see here the treatment-activated caspase-1 in some of the cells. You can notice the difference between the control population shown in red and then the LPS-treated population in black, especially in that second smaller peak. So we can clearly see a small subpopulation of brightly fluorescent cells that are beginning to appear within the LPS-treated sample. Next, we have some sample plate reader results. Again, we're looking at Jurkat cells. These cells were treated with staurosporin, and then this time they were stained with our red polycaspase detection reagent, SR-VAD-FMK. They were washed, and then they were read on a plate reader with excitation and emission settings of 550 and 590. And again, we see a strong increase in fluorescent signal within the induced population. Finally, here we're looking at some sample fluorescence microscopy results with FLICA. This image is of THP1 cells. These cells were duly stained with our FAM-FLICA polycaspase assay, as well as hooks to stain the nuclei blue. And this image shows 3 different cells. The cell in the middle is apoptotic. It has green fluorescence from the FAM-FLICA and some bright blue spots in the cell indicating the DNA is beginning to fragment. The cell on the left is probably a necrotic cell. We see no green and we see scattered blue fluorescence. The cell in the upper right is alive and non-apoptotic. It's a healthy cell. There's no green fluorescence, and we see that one bright concentrated blue spot, which is the nucleus. Now let's move on to our next product line, our Magic Red. Our Magic Red assay kits contain cell permeant fluorescent substrates that can be used to quantitate caspase 3,7 and cathepsin activity within your cultured cells. So with these substrate-based kits, not only are you able to quantitate the activity as an endpoint reading, but you also have the option to monitor in real time as well, as we'll be discussing in one of the examples. So at this time, we offer the kits for cathepsins B, K, and L, as well as for caspase 3,7. All of our Magic Red kits contain the Magic Red substrate, hooks to stain the nuclei, and then acridine orange as well to identify the lysosomes. Magic Red results can be analyzed using a fluorescence microscope or a fluorescence plate reader. Cancer research applications of our Magic Red products abound, as cathepsins have been found to be upregulated in many different types of cancers. Cathepsins are highly expressed in invasive tumors, and studies have demonstrated that they play a role in metastasis. Cathepsin B is highly expressed in colon, pancreatic, ovarian, breast, and lung cancers, melanoma, cervical cancer, bladder cancer, and hepatocellular carcinomas. Cathepsin K is activated in breast, lung, and gastric cancers, squamous and basal cell carcinomas, melanoma, prostate, and renal cancers. Cathepsin L is prevalent in breast, lung, gastric, and colon cancers, head and neck carcinomas, melanoma, gliomas, ovarian, and pancreatic cancers. Causal roles for cathepsins in cancer have been demonstrated in a variety of ways. In terms of pharmacological techniques, cathepsin activity has been downregulated through increased expression of inhibitors, as well as administration of small molecule inhibitors. Thus, cathepsins may serve as potential therapeutic targets. To get started with Magic Red, just add the substrate directly to the cell culture media, incubate, and analyze. The Magic Red substrate easily penetrates the cell membrane and the membranes of the organelles, entering the cell in a non-fluorescent state. If cathepsin or caspase 3-7 enzymes are active, they will cleave off the two targeting sequences, and this allows the crescent violet fluorophore to become fluorescent upon excitation. The red fluorescent product will stay inside the cell, and we often see it aggregating within the lysosomes and other areas of low pH. As the protease activity continues and more magic red substrate is cleaved, the red signal will intensify as the red fluorescent product is continuing to accumulate within the organelles. Negative cells, however, will exhibit very low levels of background red fluorescence that tends to be more evenly distributed throughout the cell. Ultimately, if the treatment or the experimental condition stimulates cathepsin or caspase 3-7 activity, cells containing elevated levels of activity will be appearing brighter red than cells with low levels. So let's take a minute and look at some magic red sample data. In this experiment, we have rat fibroblasts that were seated in 12 well plates. The following day, they were irradiated to induce apoptosis. Magic red caspase 3-7 substrate was added, and then the cells were photographed over the next 16 hours. And we see here in this series of images the increasing red intensity as the caspase activity and apoptosis was progressing in these cells. This next example, we see THP1 cells. They're fluorescing red after staining with our magic red cathepsin B assay. And here we see the red fluorophore concentrated inside the lysosomes. Moving on now to our MitoPT kits. These kits allow you to quantitate mitochondrial functionality and apoptosis. Detection of the mitochondrial membrane potential depolarization event really allows for an early and reliable detection of cellular health and apoptosis. These kits are very fast. Our MitoPT kits have incubation time starting at just 15 to 20 minutes. And we do offer 3 different types of MitoPT kits. We'll get into the difference between these different options shortly. But first, how does this apply to cancer research? Well, looking at mitochondrial health is important because depolarization of their inner mitochondrial membrane can be an early indicator of mitochondrial outer membrane permeabilization and the corresponding leakage of proteins that facilitates apoptosis. So mitochondrial health and function plays a central role in the health and well-being of the cell population. Because mitochondrial dysfunction has been linked to cancer metastasis and progression, mitochondria remain an important organelle of study within the field of cancer research. So here we have a little bit of background information on JC1. JC1 is a lipophilic cationic dye. It has a positive charge and lipophilic solubility. So this enables it to be readily membrane permeant, and it easily penetrates living cells. If the cell is healthy and non-apoptotic, JC1 will enter the negatively charged mitochondria and accumulate. As the dye accumulates, it spontaneously forms complexes known as J aggregates, and these have an intense red or orange-red fluorescence with a peak emission of 595. In contrast, however, in apoptotic or metabolically stressed cells, there will be fewer bright orange-red fluorescent mitochondria and more dim or non-fluorescent mitochondria. So in these cells, the JC1 will be primarily distributed throughout the cytosol, in which case it would take on its monomeric form and exhibit a green fluorescence property with a peak emission at 527. So because of this property where it shifts from green to red with increasing concentration or aggregation within the mitochondria, JC1 allows for dual color and ratiometric semi-quantitative assessment of mitochondrial polarization states. This is a nice image in that it highlights those dual color fluorescence properties of JC1. So here we're looking at Jurkat cells. These cells were treated with staurosporin to induce apoptosis or with DMSO as a negative control. And then they were stained with JC1 and washed twice. So the two cells on the upper and lower left contain mitochondria with polarized inner membranes. We can clearly see the JC1 is concentrated inside the mitochondria, and it's visible as that bright orange fluorescence. The three cells on the right are apoptotic, and they contain mitochondria that are in various stages of depolarization. So these cells appear green. The mitochondria are no longer able to accumulate the JC1 dye, and thus it's become dispersed throughout the cytoplasm. Moving on to ICT's TMRE and TMRM kits. Like JC1, these dyes also display a lipophilic solubility characteristic that allows them to be readily membrane permeant, and they easily penetrate living cells. And they also have a delocalized positive charge throughout their structure, so this allows them to redistribute across cell membranes in a voltage-dependent manner. In contrast to JC1, however, TMRE and TMRM are monochromatic dyes, so you don't see a color shift with these dyes. They selectively enter negatively charged mitochondria within healthy non-apoptotic cells, and they accumulate there where they do fluoresce orange upon excitation. However, in apoptotic cells or metabolically stressed cells, they are no longer able to accumulate inside the depolarized mitochondria, and thus they become more evenly dispersed throughout the cytosol. So in this type of situation, the overall cellular fluorescence levels drop off dramatically. And so at the end of the day, you have healthy cells fluorescing orange, whereas cells with depolarized mitochondria would exhibit lower levels of this orange fluorescence. Structurally, TMRE and TMRM are nearly identical, so they're the ethyl and methyl ester forms of the same compound. As you might expect with such structural similarity, the excitation and emission optima of the two dyes are very similar as well. TMRE has a peak excitation at 549 and a peak emission at 574, and TMRM has a peak excitation at 548 and emission at 573. So in these data, Jurkat cells were treated with either DMSO as the negative control, and this is pictured in the top panel, or 1 micromolar staurosporin for 2 hours to induce apoptosis, and this is pictured in the bottom panel. The cells were then stained with ICT's MitoPT TMRE, and then they were washed, and then differential interference contrast, or DIC, and then the fluorescent images were acquired. The top set of images show normal, healthy cells. The mitochondria are polarized, and these cells have concentrated the TMRE, so we see that visible bright orange-red fluorescence. In the lower image, we see the apoptotic cells bearing depolarized mitochondria, and we clearly see these cells are no longer concentrating the TMRE reagent, and that's what's leading to this dramatic reduction in fluorescence intensity. Shifting gears here now to talk about another product line, our cytotoxicity assays. We've now developed 2 different assays to measure cell-mediated cytotoxicity. So if you're looking to measure necrotic and membrane-compromised cells in a mixed population, sample of target and effector cells, you would use our Basic Cytotoxicity Assay. However, if you're looking to further distinguish apoptosis from necrosis, leading to a more accurate overall assessment of cytolytic activity, the Total Cytotoxicity Assay would provide this more complete characterization. So this test will reveal a population of cells that have active caspase enzymes, so meaning they are apoptotic, but they're not yet membrane compromised, so thus they would be negative if you were using necrosis detection alone. So we found this population is often in the neighborhood of 10% or more of the cells. So this can actually be a significant number of cells that other cytotoxicity assays on the market would not be able to detect. Our cytotoxicity kits provide a direct measurement of cytotoxicity, so rather than an indirect indicator, such as the release of LDH or ATP, for example. Again, as with our other kits, there's no need to lyse your cells for your cytotoxicity studies. So this makes these kits very user-friendly, easy to use, and their flow cytometry readout allows for sensitive, in-depth analysis at the level of the individual cells. In terms of cancer research, cytolytic activity is a very important process used by the body for eliminating intracellular pathogens and cancer cells. So this process is accomplished through various immune effector mechanisms, including natural killer leukocytes, or NK cells. Natural killer activity is facilitated by non-specifically lysing infected cells through the use of natural killer receptors recognizing IgG, which has been bound to antigens at the target cell surface. NK cells may also induce apoptosis in target cells. The activity of NK cells and their effect on target cells is something that's frequently studied in immunomodulation experiments. Immunomodulation encompasses all of the therapeutic interventions aimed at modifying the immune response. So that is situations where the immune response is induced, amplified, attenuated, or prevented according to therapeutic goals. So, for example, if the immunotherapy goal is to fight cancer, the immune response is augmented to help accomplish this. So first I'm going to run through how our basic cytotoxicity assay works. First, the target cells are purified and stained with CFSE, which is a green cellular stain, and it's used to distinguish them from the unstained effector cells. Next, the unstained effectors are added, and then the sample is incubated and allowed time for the effectors to exert their cytotoxic effects. 7-AED, which is a red live dead stain, is then added. 7-AED enters the cells with compromised cell membranes, so these cells are in necrosis or late-stage apoptosis, and it will bind to the DNA. 7-AED does not enter the cells if the membranes are still intact. Finally, you'll run your controls and then analyze your samples by flow cytometry, and you'll be able to quantify two populations of target cells, live cells, which are only green with CFSE, and necrotic or dead cells, which are duly labeled with both green and red fluorescence. So here we have some sample basic cytotoxicity assay results showing how this assay easily distinguishes the green CFSE labeled target cells from the effector cells. Target cells are then further distinguished into live and dead subpopulations using that red live dead stain 7-AED. Next, to expand on the capabilities of the basic test, we'll go ahead and take a look at our total cytotoxicity assay. The first two steps of this kit are the same: purifying the target cells, staining with CFSE, and then adding the effector cells. Where this protocol starts to differ is that next you add the orange-red FLICA Apoptosis detection reagent, and this reagent enters the cells and binds to active caspases during this step. Now, if the cells are not apoptotic, the FLICA will just wash out. Then again, you'll add the red 7-AED live dead stain, which, just like in the basic kit, enters the cells with compromised membranes in necrosis or those in late-stage apoptosis and binds to the DNA. Finally, you'll run your various controls, and then you'll use flow cytometry to quantify 4 populations of target cells within your sample. The 4 populations this kit is able to detect are live cells, early apoptotic cells, late apoptotic cells, and necrotic cells. Here we see some sample total cytotoxicity assay results. As with the basic assay, the CFSE is first used to distinguish the target from the effector cells by labeling those target cells green. The target cells are then further distinguished into 4 populations: living, necrotic, early apoptotic, and late apoptotic subpopulations using that SR FLICA orange-red polycaspase inhibitor probe, as well as the 7A-AD red live dead stain. Moving on now to cover our FLISP product line. Our FLISP reagents, or fluorescent labeled inhibitors of serine proteases, are cell permeant fluorescent inhibitor probes that are used to quantitate serine protease activity within cultured cells. Our FLISP assays are available with both green and red options. Serine proteases are defined by the presence of a serine residue at the active center of the enzyme. The most characterized enzymes of this type are trypsin and chymotrypsin. All living cells do have a base level of chymotrypsin-like enzymatic activity, which does vary with the physiological state of the cell, as well as varies by cell type. Activated serine proteases play major roles in several different functions. They are involved in apoptosis. In cancer, they mediate a variety of events related to tumor invasion and metastasis. They serve as markers of tumor malignancy, function as diagnostic and prognostic indicators of breast carcinomas and head and neck cancers, as well as being altered. Serine protease activity is altered in a variety of other cell-mediated diseases related to transplant rejection and infections. For our FLISP kits, once again, all you have to do is add the cell permeant reagent directly to the cell culture media, incubate, and then wash. Because of their cell permeants, they will efficiently diffuse in and out of all the living cells. If there's an active chymotrypsin-like serine protease inside the cell, it will covalently bond with the FLISP inhibitor, and this allows it to retain the green or red fluorescent signal within the cell. Cells containing lower levels of chymotrypsin-like enzyme activity will retain a much lower level of fluorescence compared to cells containing higher concentrations. So this allows you to very clearly differentiate the populations. So now that we've covered how the FLISP kits work, let's look at some sample data. This is from a flow cytometry experiment. The first panel on the left shows the fluorescence in the negative control treated Jurkat cell population. And we can see the population is negative for serine protease activity, as measured by staining with our green serine protease detection reagent. The middle panel shows the experimentally treated cells. This sample was treated with staurosporin for 4 hours to induce serine protease activity. And we do see that the majority of the cells are positive. Finally, the right panel shows the combined plot. And this is produced by overlaying these two samples. The populations are easily differentiated. This plot clearly shows the increased fluorescence that occurred as a result of the treatment. The one other kit that I wanted to mention is our necrosis versus apoptosis assay. As we've discussed, these are two different types of cell death and are both important in cancer studies as well as other cell culture studies. This kit will allow you to identify both the apoptotic and the necrotic cells within a single sample. Apoptotic cells are identified using ICT's FLICA reagent, and loss of integrity of the cell membrane, which is indicative of necrosis or late-stage apoptosis, can be detected using that 7A-AD, the red fluorescing live dead stain that we talked about. And as we saw with our cytotoxicity kits, this dye is normally impermeant, but it does enter cell membrane compromised cells and bind with the DNA. So with 7A-AD alone, you would not be able to detect the cells in early stages of apoptosis, as their cell membranes are still intact at that point, and therefore they are capable of excluding it. As caspases are active during early apoptosis, by combining the FAM-FLICA with the 7-AAD, this kit allows you to obtain a more detailed picture of the overall health of your cell population. And it can be analyzed using fluorescence microscopy or flow cytometry. So we can see here in this image that the early stage apoptotic cells are stained green. There's just a few necrotic cells in this sample, and these are only have red fluorescence. And then we do see some green and red fluorescing cells that are in late stage apoptosis as well. Our last topic that we'll be covering today is pyroptosis. Pyroptosis is a highly inflammatory form of programmed cell death, and this pathway is distinct from apoptotic cell death in that it results in plasma membrane rupture and the release of pro-inflammatory cytokines. So infected cells eventually swell, burst, and die. And then this in turn attracts other immune cells to fight the infection, leading to inflammation of the tissue. And then in a functional response, this would lead to rapid clearing of the infection. So in looking at this figure, we can see the pathway to the activation of the NLRP3 inflammasome. Assembly of the NLRP3 inflammasome is triggered by two signals. The first, known as the priming signal, begins with the recognition of pathogen-associated molecular patterns, or PAMPs, by toll-like receptors, such as TLR4, which through an interaction with an adapter protein triggers activation of the transcription factor NF-kappa B. Once active, NF-kappa B is translocated to the nucleus, where it leads to the synthesis of the inactive pro-inflammatory cytokine pro-IL-1 beta. Another potent pro-inflammatory cytokine precursor, pro-IL-18, is constitutively expressed. However, its expression is increased after this activation. The second signal is triggered by an ionic perturbation in the cell, such as the efflux of potassium ions, and this is caused by the purinergic P2X receptor, which is a ligand-gated ion channel that opens in response to binding of ATP. So once this second signal occurs, this results in assembly of the NLRP3 inflammasome, caspase-1 activation, and IL-1 beta and IL-18 secretion, triggering this inflammatory response. Deregulated inflammation is broadly recognized as one of the hallmarks of cancer. Evidence from numerous studies indicates that chronic inflammation facilitates resistance to growth inhibition, neoangiogenesis, apoptosis evasion, malignant transformation, and cancer metastasis. The level of pro-inflammatory cytokines IL-1 beta and IL-18 are significantly elevated in various types of cancers and have been shown to facilitate pro-carcinogenic activity. However, conflicting evidence suggests a duplex role of the inflammasome in cancer development, as it also has a role in eliminating malignant cells via the pyroptosis cell death pathway. So as we saw on the last slide, pyroptosis ultimately results in cell rupture, the release of pro-inflammatory cellular contents, and the activation of the innate immune response, which in turn may limit malignant cell survival and proliferation. So the role of the inflammasome in cancer is still emerging. There's currently a lot of interest in its potential development as a biomarker for cancer diagnosis or prognosis prediction, as well as in its potential as a therapeutic target to help improve cancer treatment outcomes. To detect pyroptosis, ICT offers our Pyroptosis caspase-1 assay to detect caspase-1 activation in your cell cultures. These kits include nigiricin to generate a positive control, and the kit is currently available in green fluorescence, and results can be analyzed with a fluorescence microscope, fluorescence plate reader, or flow cytometer. In this image, human colorectal adenocarcinoma cells were grown in polarized monolayers and then infected with wild-type salmonella, which was constitutively expressing M. cherry, and this is shown in red. After 9 hours, the live cells were incubated with ICT's active caspase-1 reagent, FAM-YVAD-FMK, for 1 hour, washed, and then fixed. This confocal image reveals an extruding cell that is infected. We see many red M. cherry labeled salmonella, and the infected cell is undergoing pyroptosis as we see this visible green fluorescence compared to the surrounding negative cells. In these images, THP1 cells were treated with either a negative control, and this is shown in the non-induced bottom panel, or they were treated with PMA to induce differentiation into macrophages. This is shown in the induced top panel. After 48 hours, the PMA was removed from the induced population, and it was replaced with fresh medium containing LPS, which was used to induce caspase-1 activation. These cells were then stained with FAM-YVAD FMK, washed, and then examined with a microscope. In the treated sample, we, of course, see many green cells. This indicates an increased level of caspase-1 activity happening here. In the non-induced, there's few green cells, and this indicates a low level of caspase-1 activity. To begin wrapping up this presentation, if you're looking for some more examples of our cell viability assay kits in use or any other ICT products, we do have an extensive list of publications where our products are being cited. As you can see from this publication map, our products are being used in publications by researchers all over the world. If you're looking for specific examples of our products in use, feel free to contact us. We'd be happy to conduct a publication search for you to highlight a product of interest. As you're planning out your project or working through an experiment, we do have a variety of resources available on our website to help you. You can find webinars such as this one on a wide variety of topics. Just go to our archive on our webinars page. We also have some short product demonstration videos if you'd like to see some of our popular kits in action. We periodically update our blog with helpful tips, new citations, and company announcements. So check there for all of your ICT news updates. And finally, we have extensive documentation in the form of product manuals, safety data sheets, and certificates of analysis. And these are all easily accessible from our product pages should you need them. To learn more about our products and services, please visit our website. Or if you have specific questions, don't hesitate to contact us directly. You can email us at help@immunochemistry.com. Most questions are answered within a day or even the same day. If you have a more urgent inquiry, you can call us as well. We always do love hearing how you're using our products and helping you troubleshoot your experiments.
Thanks, Tracy, and thanks to everyone who submitted questions. We have time for a few of them. Question 1, FLICA products species specific?
Well, it's generally understood that mammalian origin caspases are quite highly conserved in terms of their ability to react with their respective optimal caspase targeting amino acid sequences for these different caspase enzyme types. We have personally not encountered any circumstances where there were specificity differences between, say, a human-derived caspase versus an analogous caspase type from another animal genome, such as a mouse. So therefore, to the best of our knowledge, our FLICA products are species independent and should work well in a variety of different species.
Great, thanks, Tracy. Our second question, do MitoPT kits work in fixed tissues?
Well, excuse me. Unfortunately, our mitochondrial membrane potential products are not compatible with fixation practices, and this is because the fixation process itself does cause the mitochondrial membranes to depolarize. So with aldehyde-based fixatives like formaldehyde or paraformaldehyde, these solutions work by cross-linking primary amino groups, like those found in proteins, with other nearby nitrogen atoms, maybe in neighboring proteins or DNA, through CH2 methylene bridges. Alcohol-based fixatives, however, work by dehydrating the cells or tissues, denaturing the proteins, and then causing them to precipitate. Both of these techniques would cause the mitochondria to become depolarized. So all of the cells, if you were using MitoPT, would have a low level of staining, And unfortunately, you wouldn't be able to really distinguish much of anything based on your experimental treatments. Fixation methods, however, are generally compatible with FLICA, Magic Red, and many of our other cell viability kits. So if you have specific questions on using fixation in your experimental design, just ask. Just reach out and we'll help you out.
Thank you. And our last question today, can FLICA be used in trypsinized cells?
Sure. Well, trypsinization refers to the process of using trypsin to break down the proteins to dissociate the adherent cells from their culture vessel. FLICA is generally compatible with trypsinization protocols. It can be used in two different ways. First, you have the option to add the FLICA directly to the overlay media, and then you would complete your FLICA staining and wash steps while the cells are still adherent, in which case you would trypsinize the cells afterwards. Otherwise, you can trypsinize the cells, neutralize or deactivate the trypsin, and then you would carry out the FLICA staining and wash steps afterwards on the trypsinized cells. As far as neutralizing or deactivating trypsin, with most commercially available trypsin products, this is a necessary step as it prevents the trypsin from going beyond just breaking down these cell adhesion proteins to actually cleaving cellular surface proteins as well, which of course would result in cellular damage. So for this purpose, we normally recommend using fetal bovine serum or cell culture medium containing at least 20% FBS, as serum does naturally contain trypsin inhibitors that can neutralize or deactivate the trypsin. Now, of course, timing is important, as you need to leave the trypsin in contact with these adherent cells long enough to disassociate them, but not so long as to cause this excessive cleavage and cellular damage. So it's, generally about 5 minutes or so, but you kind of keep an eye on it as the process is occurring.
Great. Thanks again, Tracy, both for the Q&A session and your presentation. In closing, if we were unable to answer your question or you have further questions, please email us at help@immunochemistry.com. We will send our webinar recording within the week. Also, our next webinar is scheduled for September 19th. Dr. Brian Lee will discuss the redox relationship between HRP and TMB. Again, if you have any questions or if you have ideas for future webinars, please email us at help@immunochemistry.com. Thank you for joining us and have a great day.